I want to welcome everybody back to Grand Rounds for the Department of Medicine at Emory. This for Saad and Angie, this is our first one of the year, of the new academic year. And we always try and start with a, particularly whiz-bang presentation. And so it is fitting that we have two fantastic guests today. I might have hoped that we would have moved beyond COVID-19 for this academic year, but the answer is we have not at all. And so, in fact, the next two sessions will be COVID-19 related, and I'm really, really thrilled with our lineup. So today you get to hear from two distinguished individuals who I'll introduce to you in a second. We've asked them to give relatively brief remarks and really have lots of opportunities for questions. So I will be helping moderate through the chat. And so please feel free to put your questions in the chat. And I may ask you, if you're in a situation where you can do so, to unmute and ask your question out loud. And so if you hear that prompt. So to move on then. So I'd like to introduce our two speakers today. First is Dr. Angie Rasmussen, who is a virologist at the Vaccine and Infectious Disease Organization at the University of Saskatchewan. Her research focuses on the role of the host response in viral pathogenesis, with a particular interest in emerging viruses that are or have the potential to be major threats to global health, such as influenza, avian influenza, dengue, Ebola, MERS, and now, of course, SARS-CoV-2. And she's been a really important voice in this discussion in the last 18 months. Her work combines classical experimental virology and animal models with systems biology approaches to study the global response to infection and how that contributes to pathogenesis or protection from emerging pathogens. She graduated from Smith College with a BA in biological sciences, received 2 masters and a PhD in microbiology and immunology from Columbia. She did her postdoc at the University of Washington and was on faculty at the University of Washington and the Columbia Mailman School of Public Health, before moving to Canada in a well-publicized journey over the border, trying to get COVID results, testing results to make it across the border. So many of us have been also following closely on Twitter. Our second guest today is Dr. Saad Omer, who until 2019 was the most recently the William Fagey Professor of Global Health here at Emory. And at that point, left our fair city and moved to be the inaugural director of the Yale Institute for Global Health and a professor of medicine and epidemiology at Yale, at the Yale School of Medicine and Public Health. He has conducted studies widely throughout the world, and his research portfolio really focuses on both the epidemiology of respiratory viruses like influenza, RSV, and of course now, more recently, SARS-CoV-2, as well as clinical trials to estimate the efficacy of vaccination in a variety of, again, viral pathogens like influenza, pertussis, polio, measles, pneumococcus, and so on. In addition, and importantly for our topic today, he also has conducted many studies and has been a voice discussing interventions to increase vaccine coverage and acceptance. And also is an important voice in public health preparedness strategies in the response to large outbreaks. Dr. Omer is a graduate of the Aga Khan University and earned his MPH and PhD at Johns Hopkins. Facilitating our discussion today is someone who doesn't need a great introduction to this group, but is Carlos Del Rio, who, as you know, is Executive Associate Dean for the Emory at Grady. He's a distinguished professor in the Department of Medicine at the Division of Infectious Disease, as well as a professor in the Huber Department of Global Health at Rollins. He's the co-director of the Emory Center for AIDS Research, but I think, as we all know and have benefited from, has been a really important voice in the COVID-19 pandemic as well. So it's very fitting that he should be facilitating this. So Carlos, I'm going to turn it over to you again. Feel free to put questions in the chat at any point, and I'll keep track of those. And Carlos, I'll let you facilitate from here on.
Well, thank you, Wendy, and thank you both Angie and Saad for joining us today. You know, we are hoping to have a discussion here in which we'll hear about variants, we'll hear about what's happening with variants, and then talk about vaccines and talk about a topic that I've asked, Saad is one of the world's experts on, which is the whole issue of vaccine hesitancy and vaccine acceptance. And then we'll move into a Q&A session with them. So let's start with you, Angie, just giving us your presentation of where we are with variants, what's happening, why are we still having, and do we need to worry about the next variants, right? I tell people that Greek alphabet has 24 letters, and we're right now, mu is number 12, so we have still plenty to go.
Thanks so much, Carlos. And I'm going to try to blaze through this as quickly as possible. What I wanted to do is give everybody kind of a primer in viral mutation and why these variants keep emerging, so that everybody's on the same page for fielding questions about it towards the end. And so my talk is called Just Your Average Scariants, and it mostly focuses on Delta. But really, it's a primer on viral evolution, because I think the way that we've been talking about these variants is really talking about them as if they're the worst thing ever. They're some kind of super viruses, when really, unfortunately, they're an expected consequence of having the virus replicating out of control. And I'm going to talk a little bit about why that is. But first, I'm going to start with my territorial acknowledgement and equity statement. And I'm presenting today from Saskatchewan as an uninvited settler living and working on Treaty 6 territory and the ancestral lands of the Métis people. And I acknowledge and honor the First Peoples of North America and their tribal governments, histories, and ancestry, and the role today in caring for these lands and waters. And I'd also like to acknowledge the long history of systemic inequity in academic science. Two institutions where I've worked and trained, Columbia University and Georgetown University, were founded using profits from the transatlantic slave trade and excluded women and people of color from their academic communities for nearly 2 centuries. This has left a long and painful legacy of racial and gender-based inequality that continues to this day. So I encourage all of you to consider how you can contribute to making biomedical research a more equitable enterprise. And first, I'm just going to start off talking about coronaviruses. We've all been hearing about them, probably more than we like, for the last almost 2 years now. Coronaviruses are positive sense single-stranded RNA viruses. They are surrounded by a lipid envelope. This is what the virion or virus particle looks like. They actually have fairly large genomes for RNA viruses. And they actually, some of them, including SARS coronavirus 2, do have some partial proofreading capability, which is important in terms of thinking about the mutation rate. The large genome size for this type of virus, for RNA viruses, most of them are about 1/3 of this size or even smaller, means that they really do have to acquire more mutations before some of those mutations have a chance to become meaningful. And I'll talk a little bit more about that going forward. Also, I just wanted to give everybody sort of a primer, a very quick primer in how viruses replicate. So this is the standard viral replication cycle. In this case, specifically the replication cycle of SARS coronavirus 2. SARS coronavirus 2 binds its receptor at the cell surface. That's a protein called ACE2. However, in order to bind ACE2, it has to be processed by several other host proteins, including this one called TMPRSS2 and another one called furin that I'll talk about in a little bit. After binding an entry, it's internalized into an endosomal compartment. It's further processed, infusion occurs, leading to the release of the viral genome into the cytoplasm. Viral proteins are made. The proteins are then processed after translation, and the virus begins replicating its genome as well as producing subgenomic mRNAs. This genome replication process is really where all of the mutation occurs. And so that's an important thing to understand, that the more opportunities the virus is given to undergo this replication cycle, the more mutations it will acquire. And as we all know, widespread transmission in a population gives it a lot of those different opportunities. After that, the newly synthesized virus genomes are transported along with newly synthesized viral proteins to the viral surface through the cell's transport mechanisms, the endoplasmic reticulum and the Golgi. And then virions are assembled and progeny virions are released to go on and infect a new cell. So in terms of mutation, as I mentioned, this occurs at the stage of virus replication. As I also mentioned, the SARS coronavirus 2 genome is quite large for an RNA virus, but in general, RNA virus genomes are much smaller than higher eukaryotes such as us that have DNA genomes. as well as the RNA-dependent RNA polymerases that replicate the genomes don't have proofreading capabilities. So that means they have, in general, a much higher mutation rate. Now, SARS coronavirus 2, as I also mentioned, does have some partial proofreading capabilities, which decreases its mutation rate a little bit. But in general, the mutation rate is much higher than for organisms with larger genomes or DNA genomes. And what this means in practice is that a parental virus will infect a cell and begin replicating. And over the course of this replication, because of that error-prone RNA-dependent RNA polymerase, or RDRP, it will make mutants that have both defective genomes as well as mutants that have acquired mutations that may give it some type of advantage, as well as actually mutations that don't really have any impact whatsoever. Now, the mutations that result in a defective virus will, of course, be subject to negative evolutionary selection. Those that have an advantage, though, will be subject to positive evolutionary selection, and those genomes containing those mutations will go on to take over the population. Now, usually you have a combination of viruses in any given host. This is sometimes referred to as quasi-species. But you will see that with mutations that really do provide a strong advantage, they will rapidly outcompete other mutants in the population. And so that leads us to now, where we have these 4 variants of concern. As Carlos pointed out, we have 12 variants of interest that have been designated with different Greek letters. The 4 VOCs are alpha, beta, gamma, and delta. And in general, they all have different constellations of mutations. The ones that we know best and that we talk about the most, though, are the ones in the spike protein, because they determine not only infectivity, but they also have implications for how well vaccines will function. And as we've seen, unfortunately, over the course of the summer and the late spring, that Delta has become the dominant variant in the United States and really in the rest of the world. Now, it's hard to look at this globally because there's not consistent genomic surveillance. And in fact, it's even very difficult to look at this in the US because our genomic surveillance is so spotty and decentralized. But you can see here that B117, or alpha, which was dominant from the last part of 2020 up until Delta emerged, and it really emerged from the large spring surge that happened in India. You see B117 or B617.2 Delta begin to increasingly gain ground, to the point where last week it was by far the dominant variant in the US. And this has happened over and over everywhere that's had an introduction of Delta. Some of you may have heard that there are different sublineages of Delta, and we can certainly talk about that in a little bit. But in general, Delta of all varieties is pretty bad in that it's really the dominant variant right now that's circulating. So what are these mutations doing? As I pointed out, there's a bunch of different mutations in all of these VOCs and really in all of the variants. And there's a lot more variants than just the variants of interest too. There are really, every time a virus is spreading from one host to another, you have the opportunity for new variants to arise. But again, they're only going to become dominant. We're only really going to care about them if they have some sort of advantage. And for us, an advantage that we should be concerned about is increased transmissibility or increased immune evasion. This is a map here of the spike protein and all of the different mutations from some of the different variants. And this was really made before Delta became a huge, before we realized that Delta was as dominant as it was going to be. But I've circled here in red some of the key mutations that we should be paying attention to in Delta. And of course, it's worth noting that there are many other mutations throughout the entire genome, but we don't really know what a lot of those do. These ones in spike are really the ones that are best studied. So Delta is more transmissible, as we all know. This is just one paper from many that has shown that Delta is in fact more transmissible. This is data from the UK. So what's the basis of that increased transmissibility? And that's really the question that I'm interested in as a virologist. That comes down to, what are these different mutations doing? Now, you may have noticed that I've put a little asterisk here by this P681 mutation that's present in both alpha and delta. In alpha, it's a proline that's mutated to a histidine. In delta, it's mutated to an arginine. But it's important to note that both of these are more transmissible variants, and both of them have this mutation. It's in a part of the spike protein called the furin cleavage site that you may have heard of because it's important not only for this, but it's a big part of the virus origins discussion as well. And I'm going to talk a little bit more about what that might mean and what that might mean for the function of Delta. So we know that Delta has increased fitness, and I apologize for the quality of these figures. Unfortunately, preprints don't always have final figures in them, so they're a little hard to read. But this is a study that came out of China, and it's really difficult to do these types of studies. But in this case, there were people who were known to be exposed to Delta, and they were in quarantine or isolation, and they were being tested every day to see if they were going to become positive. And what they found was that all of these infections were predominantly caused by Delta. And compared to infections that happened in 2020 that were not Delta, these people had viral loads that increased faster. If they were infected with Delta, they also were shedding 1,000 times more viral RNA, which is not analogous to infectious virus, but certainly 1,000-fold increase in viral RNA definitely does indicate some type of increase in infectious virus that's being shed. Delta also replicates more quickly. So in this case, it's showing basically when the people infected with Delta crossed a threshold for becoming contagious compared to people who were infected with the strains that were circulating in 2020. So this all means that Delta basically is replicating a lot better than the other variants. Now, as I mentioned, the furin cleavage site is really important. And the reason it's important during the viral infection cycle is for entry and fusion. So as I mentioned at the very beginning, when a virus attaches to its receptor, it is taken up in the cell in an endosome, and then the spike protein continues to be processed. So the spike protein has to be processed to get the receptor binding domain into this up conformation to bind ACE2 and initiate this process. But after that happens, it's further processed with this cut here at the S1, S2 junction. And that is efficiently carried out by the protein furin for SARS coronavirus 2, which is a fairly ubiquitous host protease. Now, the reason this is important is it exposes the membrane fusion domain, which is really important both for internalization and infection, as well as potentially infecting other cells down the road. So that's really what the furin cleavage site does. And that's why mutation in the furin cleavage site might lead to a virus that is both more fit and also has increased infectivity. And that appears to be exactly what's happening with Delta. So this is a study, also a preprint from a group in Japan that looked at this in a bunch of different cell lines. And really what they found was that the furin cleavage site mutation led to increased fusogenicity or syncytia formation. This is basically large clumps of cells that form when one virus is moving from one infected cell to another. And that's what you can see here. This is a pretty low resolution way of looking at this. But basically, if you see bigger green clumps here with this GFP furin cleavage site mutation reporter, then you're seeing more syncytia forming, and that means that the virus is more fusogenic and more infective. So another question that has been on, I think, everybody's mind is whether or not Delta is more pathogenic or virulent. And this was raised for the alpha variant as well. And here you can just see two of many papers that show that infection with either of these variants, alpha and to a greater degree Delta, increases your risk of hospitalization and death. Now, this is epidemiological data, though, and it doesn't tell us very much about the mechanism of potential pathogenicity. And unfortunately, when you put this into the Syrian hamster animal model, the answer is a resounding maybe. So again, we see here that this is more fusogenic. The virus is more fit. It has increased infectivity. But when you look at it in hamsters, in Delta compared to progenitor variant or strain of the B11 lineage, you see that there is a decrease in body weight over time, which is a very crude measurement of illness and disease in these animal models. But it's not a huge difference. And it's hard to say if this difference in the real world translates to a difference from one patient to another. Also very, very difficult sometimes to take a sort of mild phenotype like this in an animal model and translate it to people, just because people obviously are much more complex and there's much more inter-individual variability than in a well-controlled animal study. So that question is still up for grabs, but I just really like to end with this. And this is a very busy slide, but really what this slide is showing is that the variants are not under immune selection pressure as some have worried. So a lot of people have been concerned that the more vaccine evasive variants such as beta and gamma are really evolving in, under pressure from the immune response to SARS coronavirus 2. And I hope it gives everybody a little bit of relief to show that that's not the case. Really, what these two papers show in their very, very busy way is that basically variants are more common when those mutations occur in the receptor binding domain. And indeed, the mutations that are most associated with immune escape, which are in the receptor binding domain of spike, are also important for binding ACE2. So it doesn't look actually as much as that these are evolving in response to immune responses. It does look more like these are evolving because they increase binding of spike to ACE2. And that's why we see this K417. This is a mutation of concern. K478, another one is this N501Y mutation. These increase binding to the receptor binding domain and ACE2. They're also important for antibody neutralization. As you might imagine, if there is in this receptor binding domain here a big antibody parked there, it's going to be very difficult for the virus to bind the receptor. If there's no antibody parked there, if there's a mutation, and that mutation confers a more strong binding interaction, that's why the antibody is not going to bind, but it's not because the virus evolved to avoid the antibody. It evolved to match up and link up better to ACE2. So, I'm happy to discuss this in more detail. I realize these are extremely busy slides, but I did want to show these 3D structures of the spike protein to really show that, this is one tiny mutation in the receptor binding domain. The spike protein is this huge three-dimensional structure, and the virus would really have to evolve at a lot of these different key residues to completely evade the neutralizing antibody response, which is polyclonal and really will attack the entirety of the spike protein. So we shouldn't worry too much about increased immune evasive mutants. My opinion is that we've seen time and time again that the more transmissible mutants like alpha and delta, or the more transmissible variants, are outcompeting these vaccine evasive / ACE2 binding mutants really every time. So what we need to think about is stopping transmission rather than worrying about a hypothetical situation in which vaccines don't work anymore. And that's the end of my presentation, but I'm happy to talk about all of this and anything that I didn't touch on because I didn't want to be talking for forever. So thank you so much.
Thank you, Angie. I appreciate it. Let's move on to Saad. And again, Saad, could you please then do your presentation?
Thank you. I'm trying to share my slides. Just give me one moment.
You got it.
Okay, good. Thanks for the opportunity. It's always good to come back and talk to colleagues at Emory, even though this one is virtual. And so I'll be focusing on vaccine acceptance and a couple of other related behavioral issues, not a lot of the other work we have been focused on in my group, like vaccine effectiveness, some of the immunological responses with collaborators and some of the other work. But the reason I think we should keep this kind of an approach, not just from our group, but others in mind, is that often vaccine acceptance strategies are thought to be something anyone can do in the sense that they are hunch-based strategies or based on our own personal experiences. I think we should keep in mind that we need to have the same level of rigor in evidence that is available for some of the other interventions and some of the other thinking around this pandemic. So let's start the story from January 23, 2020. With one confirmed case of what was then called the novel coronavirus, I wrote this op-ed in the New York Times on January 23rd. It would have come out on January 22nd, but I took a day off to actually go see Colbert in New York. That's one advantage of having a small train ride away from New York, that you get to do that. But so this was with one case, and there are 3 warnings that were included in this. And the title, as you can see, was "Is America Ready for Another Outbreak?" And the subtitle was "No, but you can do a few things about it." The first thing was that the information will change quickly, so therefore scientists should be in the lead. The second was that there should be no false assurance when it comes to communicating around this outbreak. And the third was that beware of not only the traditional sources of misinformation and disinformation, but also newer sources where well-meaning people will share their preprints with good intentions and they can be misconstrued. Unfortunately, all these warnings turned out to be prescient. So we did our first study in terms of behavioral responses to this pandemic on February 8th, 2020. And we said, we did this national study where we asked them, among other things, confidence in leadership and sources of information to see how we can better prepare for this pandemic or this outbreak. At that time, it wasn't declared a pandemic. And we asked them who should be in charge of America's COVID-19 outbreak response. That's well before Tony Fauci was a household name and people are wearing socks with his image on it. And we asked about various leaders by their title, not their names. And we found that people wanted a scientific response at the forefront. They didn't want even the president or the Congress in the lead. The other thing that remains true, and that's not a surprise, and I'll come back to that in a second, is that, what were the most trusted sources of information about the pandemic? Number one was healthcare professionals. With all this cacophony around social media and other media, it's essentially healthcare professionals, either on the media, but also in terms of interpersonal communication, that remain the most trusted source of information. And then other sources followed this most salient source. So just to remind everyone, we got a vaccine. This one, I could show any number of slides. This is from one of our studies that we conducted in the US veterans population. And we showed, amongst others, that there was really high vaccine effectiveness as these vaccines became available. So this came out in the Annals of Internal Medicine subsequently. But pivoting back to vaccine acceptance, let's look at the overall picture, the global picture. We conducted this survey, the study that came out in Nature Medicine, where we looked at 10 LSMICs and 2 developed countries, and including the US and Russia, and it had several thousand participants in total, with the country level sample size ranging from 862 through 3,362 at the country level. And what we found was that overall there was a lot of heterogeneity in vaccine acceptance. But if you look at it, overall, LMICs had higher acceptance of vaccines, COVID-19 vaccines, compared to the US and Russia. And the reason I'm bringing it up is there is an emerging trope that is out there. So this paper is out, it says on the slide in press, but it's been out for a few months now, for a couple of months now. And the idea that is being pushed is that if you make these vaccines available in low-income countries, there will be acceptance issues. There will certainly be delivery challenges, and there will even be acceptance challenges. But I find it interesting that that question was never raised. And this sort of talking point is being pushed by actually a couple of prominent people in the industry, in the pharmaceutical industry, amongst the vaccine manufacturers. But what we show, and then subsequently others show, that while there is a lot of room for intervention in low and middle income countries, the acceptance, the starting acceptance is higher than the US and Russia. We also did this, thinking about global equity. This is hot off the press, and we modeled, what is this optimal strategy if in countries like the US, after a certain level of vaccination, and the idea is, to our surprise, that is to actually have high levels of donation, not just extra doses, but actual active sort of distribution of vaccines in low and middle income countries, especially COVAX eligible countries. The one caveat when we did this work, we assumed an R naught of 2.5, but the broader lessons in our subsequent analysis hold up that a very efficient of distribution is looking after your own population, but do early global equity. And the reason why I'm bringing it up is that a lot of times we see these kinds of scenarios played out at just a national level, but we are truly in this together, and vaccine acceptance and delivery go hand in hand, and that shouldn't just be focused domestically, but also globally. In May 2020, going back to that time, before the vaccines became available, we did this national survey, and we found that there were not only suboptimal vaccine acceptance in the US, we also developed this model that predicted that it will be differential by age, so younger individuals will have lower vaccine acceptance. There will be gender factors, males will have lower acceptance. There will be regional heterogeneity. There will be racial disparities, and there will be educational disparities as well. So things that eventually were determinants or factors in vaccine acceptance were out there. There were signs there as early as May 2020. But again, in our global work, going back to some of that work, this is unfortunately a little bit small, but it says health workers or healthcare providers, and the second one is Ministry of Health. These are the people, if you look at across countries, which of the following people should you trust most to help you decide whether you would get the COVID-19 vaccine? So essentially, the influencers remain consistent, the remarkable consistency of healthcare providers across countries. There may be differences between countries in trust in healthcare providers, but they remain the most significant group of people where you need to focus on in terms of delivering vaccine confidence interventions. We also were looking at in fall 2020, early fall 2020, what would happen if the vaccines were approved attached to the political calendar. And there was a lot of discussion there. And we had this online experiment with my colleagues in social sciences. And we found, we had the control group of an authorization of the vaccine, of the COVID-19 vaccine, the first authorization, a week after the election. And we intentionally anchored on the election. We gave them both the date and the election date. And we said, compared to that, if the approval happens 1 week before the election, there will be lower confidence and lower uptake. So trust in the system is important, and that continues to be relevant. We also found that December 10 approval, that's the date we projected of authorization, etc., if things take their normal course, will result in higher confidence, but not necessarily higher uptake. But so we were, the fact that career folks in FDA and others stood up to a lot of pressure, and this is not a political statement, that's documented, that's on the record, actually, I think facilitated some initial confidence in vaccine acceptance. We also said, what are the major influencers on vaccines? So we randomized people to have positive statements, and we did this online experiment that someone says they believe that vaccine is safe and effective, versus negative statement, they don't believe that vaccines are safe and effective. The bottom line is Dr. Fauci had the most salient impact. And we should remember that we are in this plateau, but there was a lot of progress, and that some of the initial phases, having these statements did help, at least according to our data. And this was before the vaccine became available. We did this experiment. His negative statement would have doomed the vaccine. And we also found that political leaders, national leaders, if they have a bipartisan stance, that was effective, even amongst Republicans as well as Democrats. But if they had different things to say about the vaccine, it was not a negative impact, but it was a muddle, and you didn't make much progress. Then we have been doing online experiments for different kinds of messages. So for example, we did messages around bravery, and we showed that compared to our control, if you frame, and this is a longer vignette, but if you say you're not brave if you don't get COVID-19 vaccine, you have an improvement in intention to vaccinate yourself, but also advising others. The other thing we found was community interest. So getting the vaccine reduces the risk of spreading the disease to others. And then this is an experimental outcome. We are not using it in the field, but we wanted to test the boundaries of this messaging. If you say that community interest with a experimental message for embarrassment, imagine how embarrassed you will be if you don't get the COVID vaccine and spread it to others. That also moves people, but it has other side effects, etc. So therefore, we don't advise it to use it in actual messaging, but this was an experimental message. Here's the other thing, now these kinds of messages are being applied to a large sort of state level trials that we are setting up, where we are incorporating these messages into face-to-face persuasion that some of the community organizations are doing. So we're doing a cluster randomized trial. In July 2020, we thought that we should be ready for a discussion around vaccine mandate. At that time, it wasn't part of the national conversation, but we knew that there may come a time where it would be needed as a policy option. So we crystallized a few decades of work on this topic to say that there should be 6 trigger criteria for this vaccine to be mandated. So it should be inadequately contained. It should be recommended by, the outbreak should be inadequately contained, should be recommended by the ACIP. There should be adequate supply. There should be transparent communication. The infrastructure for delivery should be present. And most importantly, there should be inadequate voluntary response. Based on this framework, and obviously this was shared and was accessible to policymakers, right now, we have reached these criteria. We have met these criteria for an overall for a selected mandate for employers and healthcare workers, but not necessarily as a generalized mandate. So I'll sort of delve into some of our values-based work. So values postulated as that we have some social psychologists postulate that just like we have taste buds that give us combinations of flavors based on combination of stimuli, we have moral taste buds, 6 universal moral taste buds, but they are emphasized differentially in different people. And so we did, back in 2017, we did the study that for the first time showed, that was around HPV, that for the first time showed that vaccine decisions can be moral decisions. So those who emphasize authority are more accepting of vaccines. Those who emphasize purity in their overall value framework are more hesitant to vaccines, and same with liberty. And liberty and purity are the narratives that you will sense, that I don't want toxins in my body. It's my choice. That is part of the narrative as well. So what we have done now is to have done these large 5-country trials, and you will do a second phase of this with 27 campaigns that are evaluated in the randomized model across these 5 countries with tens of millions of subjects and a sample size of tens of millions, and it varies depending on countries. And what we found was, and these are very initial results, that the context matters. So local context matters, and mix of images and illustrations work. And content tested, we tested it in multiple languages, and we almost invariably found that the local languages are more effective, way more effective in persuasion. The other thing we said, what we found was that, we were able to move people across these countries. These are as varied as Ukraine and India and Pakistan and the Philippines. And we also found very specifically, and the reason I was bringing up the values-based messaging, that speaking to people's values was the most effective way of messaging on social media. The next step is we are actually going to be doing cluster randomized trials to focus on moving the actual coverage, not just attitudes, intention to vaccinate, but actual coverage measured by local systems. So I will, quickly go through the lessons here. We find that partnership works. Language matters in these delivery mechanisms of interventions. Values-based messaging shifts attitudes, and context is key to moving the needle. So I will stop here and would be happy to take any questions.
Thanks, Saad. This is absolutely terrific. Again, you've worked a lot around hesitancy, and you worked a lot around strategies to address hesitancy. We have over 80 million Americans who have yet to be vaccinated. And, do we go down the route as President Biden did of mandates, or is there anything else we can do to increase uptake of vaccines at this critical moment?
That's a really good question, Carlos. I think, we as a country have done the right thing, but only 3 to 4 months late, and it's very late in a pandemic. I'll start with an example. We should have been ready with a communications campaign in November 2020. We should have been ready for, we thought, meaning sort of the administration and others thought that we can town hall our way out of the hesitancy issues in, for example, minority populations, etc. And that was helpful. And that was essential, but not sufficient. And similarly, mandates, designing mandates, I think we are a couple of months late in implementing them. And so setting that context going forward, a few things should be there. First of all, create a benchmark for states and jurisdictions to say what are effective mandates. For example, CDC's public health law program should have data on this available, what mandates are working, what are not working, what people are doing. So we know that in state level childhood vaccine mandates, that was incredibly helpful. Second, we should have equitable ways of coming up with vaccine verification requirements. The CDC has 361 powers to actually do that, but also the Office of Technology, the Chief Technology Officer in the HHS can also do that. But we are not doing these, we're not empowering local jurisdictions to actually have equitable and effective ways of verifying immunization. And the last thing is, we should have a national CME program for healthcare providers based on evidence-based approaches to increase vaccine uptake in the clinic. And so these are the 3 things we should do pronto.
Thank you very much. Before I go to the next question from Dr. Stevens, I just want to mention to the audience that we're not talking about boosters during this session. It'll be next Tuesday at Grand Rounds that we'll be addressing boosters. So we'll try to limit questions around boosters. Dr. Stevens.
Well, actually, Carlos, that was part of my question, but first I want to thank both Angie and Saad. Good to see you again, Saad. And thank you for those great presentations. Really a question for Angie about what's next through the door, if you will, in terms of variants. And there's the report from South Africa, the new variant. We still have Lambda and Mu that are out there. What's your thought about what is next through the door, and what might we expect from a variant story? And I'll not ask Saad about boosters because he can. Sounds like that's for later.
So that's a great question. And unfortunately, it's one that, your guess is as good as mine in some ways. This is, I think, one of the real challenges of dealing with all of these variants is that when one comes out, we can do in vitro assays. We can, if we have time and the resources, we can start infecting animals and see what happens. But it's really, really hard to predict how these variants are going to perform actually in the human population. And right now, with the limited data that we have, I'm not as concerned about the so-called new South African variant, which is actually a collection of viruses. It's an entire lineage of new variants that have emerged. And I'm not worried as much about mu, although it's certainly one to watch, because none of them have that mutation in the furin cleavage site. And this is still such a limited area of information. We don't know that is the end all be all mutation that is going to make a variant outcompete the others. Certainly there have been localized or regional outbreaks of Lambda, for example, that have been really bad. But once Delta gets in the mix, it's still outcompeting them. But unfortunately, even though there's a lot of work that people are doing, and I'm not a modeler, but there's a lot of modeling work to look at the mutational space of the spike protein and try to figure out and predict what these variants are going to do. Unfortunately, the best way of figuring that out is to actually wait and see what happens. And unfortunately, even more unfortunately, doing that basically means that then we end up with something like Alpha and now Delta on our hands, where transmission is out of control and we have to try to respond rather than prepare for it. So the best way of dealing with this, in my view, is to never find out what happens with these variants, because when one emerges that we're interested in or that we think might be bad, we should take extra precautions to make sure that it doesn't spread out of control. And that, is not what has happened, unfortunately. So, I mean, I'm always really torn about this, because on one hand, there's, oh my God, new variants. And everybody gets really worried about it. And Eric Topol famously coined the term scariant. I think that's a good way of describing it, but it also runs the risk, I think, of minimizing the fact that we should be paying attention to it. By saying to people, don't freak out, is different than saying to people, don't worry about it at all. And I think that the conversation often is one of those two extremes. It's either panic or pandemic's over. And neither one of those is the right thing to do. The right thing to do is to continue enhancing our genomic surveillance, improving the way that we're tracking, improving testing. I mean, in the US, we still don't test enough, and we never have. So we don't even really have a good idea of which variants are emerging as they emerge. So it's a real challenge. And I wish that I could say, oh, we better watch out for mu, or we better watch out for C14, I think is the South African new lineage that's emerged. But I don't know, it could be something that's not even on our radar yet. Hopefully, we'll be able to distribute vaccines more widely, and we'll actually start to improve our mitigation efforts, so that Delta will be the last. But, I'm, as much as I try to be optimistic, I'm also realistic, and I don't think that's going to be the case.
Thank you.
We had a question from Dr. Kushrestha. Sorry if I butchered your name.
Thank you, Wendy. I know that Umbar is fine. Great talks. I wanted to ask about, the mention of communication, like transparent communications. We know that, science goes a little bit back and forth, with one study coming out with one finding, and then the next one comes out with somewhat different findings. So how do people at the policy level, if you put scientists in the lead, try to communicate, consistently about, findings, because things change? And then how, which I think, sometimes when you go back and forth, it actually leads to more mistrust in the public, as, like people have about CDC right now. It's like they keep saying varying things about mask and why it was lifted in the beginning of summer. So how does one reconcile this?
That's a really good question. Look, people, and we have studied and others have studied this, is people are actually tolerant of changing guidance, but what they are not tolerant of changing rationale. And so communicating what your strategy is, communicating in low salience situations, people get confused more easily by changing guidance. But this is a high salience situation where people are actually paying attention. So people do get the nuance, but you need to communicate the strategy. Is it reduction in transmission? Is it reduction in mortality? Is it reduction in social impact of the outbreak? And if it is all of the above, what is the hierarchy? What has happened is it's not that the outbreak has changed and people have responded. Unfortunately, the rationale, a lot of people are unsure of the rationale for some of these recommendations. So that would be my recommendation. And I'll give you a positive example. And I serve on the COVID-19 vaccine working group for WHO. And so I have I can see how the sausage is made and then how WHO communicates. WHO has been reasonably consistent in the rationale, and especially outside the US, because the US focuses on the CDC. These recommendations have been well received, vaccine-related recommendations. Frankly, some of the other recommendations that were communicated earlier on had this element of confusion because things have gone in directions where they have followed the outbreak, and people understand that you need to change when you're following the outbreak, as long as your strategy is consistent.
Thank you, Saad. That was a great answer. Thanks.
Thanks.
All right, I wanted to actually stick with you for a second, Saad. Dr. Wu asked a question about the use of the word mandate versus requirement for vaccines and the negative connotations that are associated with the word mandate, and your thoughts there.
That's a really good question. So I'll clarify a couple of terms. First is compulsory vaccination, where you actually force people to get vaccinated. The second is mandates / requirements, where you take away a benefit, etc. You're not physically picking up people and taking them to a vaccination center. These words are used interchangeably, requirements and mandates. I do that interchangeably. The advantage of using requirements is that it is less charged. You're absolutely right. But the advantage of using mandate is that it's more well understood. So, if you're trying to communicate a policy message where you're trying to move things, it's often okay, and it's the lesser of the two evils. For the sake of clarity, we often use the term mandate. But if you are trying to implement something where you think, in terms of communications, you'll be using a less charged word, then it's okay to use requirements, and we do that all the time.
All right, Dr. Collins, you have your hand raised.
Hi, thank you. I had a question regarding whether you have any advice on how to advise patients who are still hesitant about getting the vaccine, especially in the light of people who they know still getting COVID who are vaccinated, and if there have been any studies on that yet, just vaccine hesitancy related to people having mistrust of the vaccine now that people are still getting COVID.
So we have done some work there, and we have new sort of paper coming out on specifically on males with a conservative bent. We try to remain nonpartisan in the work because it does disservice to messaging, but sometimes you have to look at that. And we actually find that the same messages delivered effectively work that you deliver to other populations. We actually tested different kinds of messages. Without going into the detail, in the interest of time, I think I'll take the liberty of directing you to an evidence-based CME module. Actually, it's 3 modules within sort of a CME program that we developed at Yale. It is available to anyone, and that specifically focuses on clinicians and brings together the evidence around this and says, how do you deliver? There's a vignette there as well that you can observe, and so on and so forth. So it's a 55-minute CME program that anyone can take. And so I'll take the liberty of guiding you to that rather than going through the specific details of how to do that.
Thanks, Saad. Andrew, we had a question for you. Could you comment a little bit on, I guess, first off, if there's a virologic reason that we're seeing so much more disease with Delta in children, and then the impact of disease in children on the school decisions and that the whole challenge with returning to school right now?
Yeah, so in some ways, this is really more of a question for an epidemiologist, but I'm gonna take my stab at it. Anyways, knowing what I know about Delta. Now with kids, there's not really any evidence that Delta is more specifically virulent in children. It may be, as I discussed in my talk, more pathogenic in general, but again, that's still not well understood. What is understood, though, is that it's much more transmissible, and that does include children. And I think one of the most confusing things for a lot of people, and especially a lot of parents, is this incorrect idea that because children don't get sick or severely ill from COVID-19 as often, that they don't get it at all. And in fact, children can be just as easily infected with SARS coronavirus 2 as adults, and they can spread it to one another. And so when you have a variant that's more transmissible in the adult population, of course, it's also going to be more transmissible in that pediatric population. But there's also one big difference between adults and children under 12, and that is that the children under 12 are a largely naive population. And we don't really know the prevalence in kids because infection can often be asymptomatic. So we don't even really have a good idea of how many kids might actually have natural immunity. And really what that means is that, a virus doesn't think. It's essentially a little biological machine that's programmed to just make more viruses, and it's going to follow the path of least resistance. And if there's a large population of susceptible hosts in the form of children who are gathering in an environment that's conducive to transmission, like schools without mitigation measures in place, that is going to lead, unfortunately, to more outbreaks in schools, and it's going to lead to more sick kids, just because then you get into effectively a numbers game. If you have more infected kids, you're going to have more infected kids getting sick, even if it's not the same proportion as infected adults. So, my opinion, and this is what I'm advising all of my friends who do have kids under the age of 12 and family members, is that if in-person school is going to occur, it really, and there's many, many good reasons why it should, then it absolutely has to happen in an environment in which there's strong mitigation measures in place and multiple mitigation measures. So improving ventilation if possible, wearing masks, and getting vaccines available for kids at least over the age of 5, and ideally all kids as soon as possible, and also making it easy for parents to get their kids vaccinated when those kids become eligible. That might mean vaccine mandates, and I would definitely defer to Saad as far as the policy implications and the feasibility of something like that. Although it's worth pointing out that we've had vaccine mandates in schools for a long time for certainly other vaccines. But I think that, for in-person school to occur and to minimize the risk, not only to the kids, but to the larger community and to those kids' households, it's going to be really crucial to apply multiple mitigation measures and really as many mitigation measures as possible.
Thank you, Angie. Well, we're at the top of the hour, so I want to thank you and Saad for your presentations. This was excellent. Clearly, we are at a very complicated time right now in our pandemic. Delta is spreading widely, and it is not slowing anytime. And I think as more people, and we got to get more people vaccinated, and having, we're about close to 60% in the US, which is still way lower than where we need to be in many places, like in the South. We're actually lower than 50% fully vaccinated. So the more people we get vaccinated, the more we control this virus. Thanks again for your time, and continue your good work. And we'll see you everybody next week when we'll talk about boosters, and we'll talk about global equity.